Okay, so yeah, welcome again, and very warm welcome to a next session, Talking 3R. Today in the webinar, we will have Björn Gerlach with us. He's currently also affiliated at the Central Institute of Mental Health in Mannheim, and is also working for PASP, a company in Heidelberg, for good scientific practice and preclinical research. And his topic today will be about the quality system in preclinical research. And I'm very happy to have you here to speak about it. A few housekeeping rules for today. So you may have noticed, for those of you who are new to the Zoom webinar tools, so you can't unmute yourself. So in case you have any questions, there's at the bottom you have this F&A button. So in case any question arise regarding the talk, please feel free to type it in there, and then we will read those questions and answer them. In addition, Björn and Silke also prepared several questions for you before the talk and during the talk. So I would please ask you to join in for this interactive webinar today, and that we will hopefully have an intense discussion. So yeah, with that, I will hand over to Björn.
Thanks a lot, Markus, and yeah, thank you for having me here. I'm going to share my screen to have the presentation hopefully there soon. So according to my picture, you should be able to see it. Yeah, wonderful. Okay, so the focus today is on the EQIPD quality system and its characteristics and how it provides support to increase data quality. And I will also try to make the connections to the 6R during the presentation. And the idea is that we will present all questions right now. So there are in total 8 questions you should answer. And then during the presentation, then some of the answers will come at certain points. So please start the questions.
And then here's the last set of questions.
Okay, thanks a lot for taking part in the questions. Then I will start the presentation now. So before I start, I just briefly want to disclose my affiliations. I'm co-founder and shareholder of PASP GmbH and PASP US LLC. I'm employee of PASP GmbH and the Central Institute of Mental Health, so like Markus mentioned already. And I'm also founding member and member of the executive board of the guarantors of EQIPD Society. So let's get into the topic. I would like to start with this simplified cycle of research, where we have the discovery of research findings on the one side, and then also the confirmation and falsification by other research groups of these findings. And this is the self-correction process in science. And recent publications showed that something is wrong here, or at least that this process is slowed down. And we cannot go into all of these publications. You might be aware of them anyway. But I want to highlight just one of them, because that is really the one that is my main driver. And I find it really disturbing almost, because it has such traumatic effects. And this is the example from ALS research, or amyotrophic lateral sclerosis, which is a severe motor neuron disease. And here you see the changes in survival rates of the mice in this ALS model. And they were treated with these substances. And these observations, these increased survival rates, these observations were published. And not very surprisingly, based on these observations, there were then clinical trials started. And these clinical trials, they were negative. Maybe not too surprisingly, because of this translational aspect, so that animal models will not translate efficiently and so on. But then an institute in the US, the RLS-TBI Research Institute around Steve Perrin, published this study, Make Mouse Studies Work Again. And what he did is he repeated these studies with increased measures for data rigor, and meaning that he increased sample size, he introduced blinding and randomization. And what he found is that none of these previously positive substances tested positive again. So here we treated patients based on insufficient preclinical evidence. And that's, for me, again, quite disturbing, and really the motivation and driver to work towards better data quality. And so what can change to improve this self-correction and accelerate it? And here the idea is to introduce a smaller cycle to strengthen the internal rigor of the research data. And and filling this cycle with life, that was the task of the EQIPD consortium, which aimed to establish a systematic approach to provide such support. And this consortium was mainly in Europe, because it was funded by the Innovative Medicine Initiative, which is a joint partnership between the EU and EFPIA, which are the pharma companies in Europe, or an organization of the pharma companies. The project ran from 2017 to 2021, and consisted of 10 academic partners, 12 pharma companies, 8 SMEs, like also PASP. That's how we contributed there. But then also over 100 associated collaborators and stakeholders. And also on the advisory board, there was also Glenn Beckley, and from who published this first article from Amgen, that they couldn't reproduce data, and also Shai Silverberg from the NIH, but also Javier Goulin from ALEC, the European representative of ALEC. So they all contributed to this project. And there were several topics that the consortium worked on, but I only want to focus here now on the one that we will discuss today. And that is, first of all, the EQIPD quality system. That's where we will have a deeper dive into, and also briefly mention the EQIPD online platform, as well as the EQIPD certificate. And then finally, the non-profit association, the guarantors of EQIPD, to provide a sustainable system for the EQIPD quality system. And hopefully in the end, we will really improve this whole cycle again. So let's have a look at the quality system. In the center of the quality system are 18 core requirements, which apply to the interested research units. And they are broadly applicable and describing kind of minimum expectations to be implemented. And they go beyond biomedical research, but are still precise enough to be specifically tailored to research units. These 18 core requirements, they were developed in a Delphi process, meaning that a group of experts from universities and industry named requirements that were important to them. And this initial list, this first list, had over 120 items. But then by ranking them, by voting on them and setting thresholds, discussing the ones that are below the threshold, and repeating this cycle, the list was really distilled down to these 18 core requirements. And participants of this process were Uli Diernagel from the Charité, Malcolm MacLeod from University of Edinburgh, Thomas Steckler from Janssen. So really quite a broad spectrum on experts. And this concept was published last year in eLife, and I've included here small QR codes, so to provide that you have fast access, easy access to these resources, and I've also made that on a few other occasions. And the difference to existing guidelines is that this quality framework includes feedback loops that can indeed make a difference towards quality, since this is a well-known concept in many biological systems, but also in other quality systems. So there it really differs from all the guidelines that are currently published. Let's dive a little bit deeper again. It's possible to categorize these 18 core requirements into these 3 sections that enable good research practice, that establish good research practice, and maintain. And they are unequally distributed between them. In the first category, to enable, these are 3 core requirements that shall ensure responsible conduct of research and also compliance to applicable law. And this first core requirement I want to present here reads like: the research unit must have a procedure to act upon concerns of potential misconduct. Because what we often observe is that in almost all research units or research organizations, such measures are established, but usually the researchers are not aware of it. And that's kind of this first question I asked. So maybe you can show the answer to the first question to see what the responses were. So it's 50-50 basically. So yeah, there's one group knowing of it and the other group not. So all EQIPD would then request here to be compliant with this core requirement is that these 50% not being aware of such measures against potential concerns of misconduct, that they are informed basically about it. And with that, I want to come to the next core requirements. And these are 9 core requirements establishing good research practice. This is about the data itself and about the integrity and rigor of the data in respect to the design, conduct, analysis, and reporting. And what we often see in research labs here is that the implementation of protocols, that they are with the individual researchers. But once these researchers leave the lab, the protocols cannot be fully understood anymore, or some specific knowledge is missing. So, and that was then the question number 2 here. Okay, so the kind of the majority towards 0 would say 0, 1, 2, it's kind of easy. But I think that here is the possibility to improve the availability of protocols. Okay, so and after implementation of the quality system, a central database, for example, could be established to retrieve these protocols from these databases so that, yeah, they are available. And to even top that up, it would even be possible to have protocols double-checked so that somebody else has a look at them and checks whether these protocols are complete and are kind of compliant with any existing standards. I want to have here for this establishing good research practice another example. And that reads like, investigator must declare in advance whether a study is intended to inform a formal knowledge claim. And this might sound complicated, but actually it's quite easy. So the idea behind this, that when planning a study, one must decide what the results shall be used for. Are the results for exploratory purposes, so meaning that the study only shall follow a brief idea, then less rigorous measures might apply. However, if these results have the intent to be used for something bigger, like claiming new knowledge or confirming a previous study, then additional requirements must be applied to reduce any bias. Or in other words, from a Nobel Prize winner actually in physics, it was Richard Feynman. He said, the first principle is that you must not fool yourself. And you are the easiest person to fool. And that's kind of what we want to prevent here. And we will also come back later again to this little citation. So what's exactly behind it? What are these measures that must be considered here? This is really nothing crazy. And actually, in fact, they are kind of similar in the ARRIVE guidelines. And this was another question here. So how aware are you about the ARRIVE guidelines? And I guess the scale is again the same. So 0 being very aware, and 5, what are the ARRIVE guidelines? So for some of you, I guess this concept is really very well known then, and others, might want to look into the ARRIVE guidelines, because I think they are absolutely crucial for animal research. So, but let's look at this table now. So what are these measures? We cannot, of course, not go into all of them, but just very briefly. So these measures are about predefining a study plan and formulating a proper study hypothesis, thinking about blinding and randomization, and also the sample size calculation should be performed, and so on. And when performing now exploratory research, then these measures are recommended to be implemented. However, when going one step further and performing knowledge claiming research, then these measures are required. Well, in the case of blinding and randomization, then at least they are recommended, and it has to be justified why there is an exception and why that cannot be applied to a certain experiment. And this concept is absolutely crucial to improve the value of research data. And it should provide really, on the one hand, this rigor, but also on the other hand, with exploratory research, kind of a flexible approach that not all research needs to be in this narrow window. Now I want to go back to this pyramid we had before here and to come to the top, where 6 core requirements are there to maintain good research practice. And this is about the continuous improvement, or introducing some feedback loops to look into the status quo of a research unit. And this concept is based on the conventional quality systems. However, EQIPD is here very much tailored to the experiment and to the research data. And this example I want to present here reads like, risk assessment must be performed to identify factors affecting the generation, processing, and reporting of research data. This again might sound a little bit abstract, but also it's very easy again, because it's mainly about asking, when planning a study, what can go wrong during the conduct of the study. And just a brief example from my cell culture experiments, which I performed several years ago during my PhD and postdoc. When I planned experiments, I was always asking myself, which controls do I need to include? Do I have everything ready to perform the experiment? And also, what can go wrong, or what bias can I introduce during the study? And in the end, I actually found out that when preparing my samples for Western blotting and doing the immunoprecipitation, I always treated the unstimulated sample first. And that actually had an effect then on the outcome of the experiment. So in the end, I randomized my immune precipitations. But the good news here about the EQIPD quality system is that there doesn't have to be a formal process behind, especially not for such simple experiments, because it's enough for the quality system if this concept is understood and if kind of this informally approach is in place, as I just explained it. It can certainly look different with a formal process when performing larger studies or animal experiments. So here EQIPD would recommend to have in the study protocol kind of a little risk assessment table that kind of summarizes or at least points always towards some common risks and what kind of measures could be implemented against these risks. So with that, I want to come back to another aspect of the quality system, that it needs to accommodate kind of specific requirements. And why is that important? Well, there is research done all around the world, and there are many rules for the specific research units. So we kind of need to introduce some a flexibility to accommodate that. And for example, a research unit collaborating with industry might have specific requirements, or being funded by the DFG or the MRC. So then EQIPD provides this systematic approach and help to also think of these specific requirements. And there's also some guidance and help provided by EQIPD. The next aspect I briefly want to mention is the information that we created within the EQIPD consortium. And here we created online information, or the toolbox, that shall help researchers to find information and also practical information on how to implement certain aspects of good research practice. And just very briefly, here is the link to this website, and you land on the starting page, which also provides video for easier access, and then the toolbox that is a very comprehensive list of information on various aspects. And in this context, we asked the question number 4. So how accessible are information on good research practice? Was this exercise here we did nonsense, or might it be helpful? Okay, and thanks for the feedback. So there could be more information apparently provided. So then if you are not aware of this toolbox, maybe have a look at it. Maybe that you also find it helpful for your own research units. Now I want to, after going through all the aspects of the EQIPD quality system and before proceeding, I briefly would like to make the connections to the 6R principle that was published by Daniel Stresch and Uli Diernagel. And as you know, it expanded the 3 Rs by robustness, registration, and reporting. And as you have seen, EQIPD is mainly about creating this robust data, specifically in the context of knowledge claiming research. And here it clearly supports the 6 Rs by providing support for the robustness, but also pre-registration is connected here within the equality system. And also the last point, the 6 Rs, we have mentioned the protocols, that protocols need to be available. And on reporting, there are actually another 4 requirements. So EQIPD is heavily driven towards these additional 3 Rs here. But what about the others? It's also about reducing animal research. And that's because it increases the research data. It increases the quality of the research data. And here I'm thinking, for example, of Alzheimer research. I guess you might have heard that just last week articles were published that the entire role of A-beta 56 in memory of Alzheimer animal models was most likely due to fake data. So if these data were also rigorous and fulfilled these additional, fulfilled the EQIPD requirements, then that would also save lots of money and also reduce the use of animals. Now I want to switch gears again back to the quality system, and this last point here, after presenting all the information about it, because then there's also the possibility to get a certificate. And currently there are 7 research units certified and 5 of them are displayed here. It's just that these biotech companies didn't want to be on this website. And just very briefly, so for this certification, there is a brief assessment done that will normally be remote, where an interview takes place and these measures that are acquired, adapted in the research unit will be discussed and looked at. And after this assessment, there will be then a report summarizing it and then also making suggestions for any improvement if it's necessary. So with this assessment, it kind of has the purpose on providing another feedback loop that you can reflect on practices, but also provide the acknowledgement, the reward that these practices are established. And with that, I briefly want to talk about the ZI, because here we also want or aim for this certificate. So the goals at the Institute for Psychopharmacology at the ZI is most importantly that we want to ensure that all data are stored and archived according to the 2 data integrity principles, which are FAIR and Alcoa+. So FAIR meaning findable, accessible, interoperable, and reusable. And that shall be for all research data. And the other data integrity principle, Alcoa+, that is promoted by the WHO, World Health Organization, and is a bit more specific on the data. For example, it says that the data should be attributable and legible. So everybody should be able to read it and understand who created that data. Yeah, and in this context, we also plan to have a dedicated space on the institutional server for raw data, so that these raw data are kind of saved in a way that they cannot be deleted and modified anymore, so that these data are safe and that we achieve here the highest possible or the ultimate measure for data integrity for this raw data. And to kind of also support this data integrity aspects, we plan to implement an electronic lab notebook. And here we have decided for eLab FTW, which is an open source software. I guess some of you might also know it. And yeah, this will support the organization of all experimental information. So such as organizing the study protocols, organizing analyzed data, but also creating a comprehensive database for chemicals, for antibodies, but also having the TVAs in there so that it's directly possible to link them to the experiments, to the studies. And with that, we kind of hope that we also create the incentive for the researchers working with it, that they have easy access to the information and like this, provide a good incentive to use this electronic lab notebook. Because this is usually a long process, as I also know from colleagues from Berlin, from the Berlin Institute of Health, that adoption of a quality of a lab notebook just takes time. And all these efforts here at the ZI shall also ultimately receive to the EQIPD certificate. And but ultimately, and that's kind of the summary, we do not only want to implement the 6 Rs, but really live them. And if I understood correctly, then Marcus, I don't know if you also want to say something right now or maybe later. There's also the possibility to support other research organizations within Baden-Württemberg on this way towards the EQIPD quality system or on to the 6 Rs. Just very briefly, and also having the time in mind, I just want to make a few comments on this governance structure, or who actually controls, or what's the control mechanisms for this EQIPD quality system. And here we have on this strategic level, on the highest level, founded a non-profit association here in Heidelberg, which is named the Guarantors of EQIPD, and they shall have kind of the oversight of the quality system. So they shall ensure the governance and management of all tools that are associated with the system. They will also provide trainings in form of summer schools and seminar series, and also establish a stakeholder group for active engagement. And then this strategic level will work together with the operational level. And this is currently the PASP network. So this operational arm, that's the organization then that will do the assessments in the name kind of EQIPD, but there will always be the connection between the two of them, and also the reports will be double-checked then. So we'll have a feedback loop from the operational level to the strategic level. But then, of course, there's also the community level that will interact with the other partners. So with that, I briefly want to summarize. So I've talked about these 18 core requirements, and that they are there to systematically improve research and improve research data. And I hope it also became clear that they will support kind of the 6 Rs, or at least the 4 Rs as discussed. And then there is a certification as a reward to demonstrate high quality processes. And on the last slides, we have this, the guarantors of EQIPD to ensure the sustainability of the quality system. And with that, I want to thank you for your attention, and want to also provide some resources here in case you want to connect with me or just stay informed about this topic. So we publish this EQIPD Pass newsletter bi-monthly, or also publish aspects on social media. And I don't know, are there any questions at this point, or shall we directly go to the last 4 questions that we asked at the beginning? Is there anything urgent right now about the quality system?
So at the moment, I don't see any questions.
Okay. Well, then I would just continue here with this a wonderful picture, with which I want to go back to the citation from Richard Feynman. You must not fool yourself. And I'm not sure how it's full, but I can tell that always when I look at this picture, I'm certainly fooled, because no matter how I look at it, the lower moon just looks smaller, or the bigger moon, the upper moon, bigger than the lower one. So it's always depending on the circumstances, and even with the best intentions, knowing that these 2 moons have the same size, it's sometimes just quite easy to be fooled. And another example I tried to make here was the one about geography and about Barcelona and Tokyo, because always when I think about Barcelona, I think of summer, sun, beach, and thinking of Tokyo, rather a green city where the cherry trees flower. And I'm curious how you think about these 2 cities. So maybe we can have a look at the last set of questions. Oh, well, yeah, very good. So most people actually said that Tokyo is further south, which is true. So I think Tokyo is about 6-degree latitude further south than Barcelona. So then I'm not entirely sure how the second question then here is displayed. How many experiments have you pre-registered so far? Because I only see here a blue bar, 23, 27.
There are a lot of 0s and 2s and 1s. Okay. And then we have 110, 30, oh, wow. And it goes up to 50. This is the highest value.
Oh, wow. Respect. Very nice. Yeah. But so what I hear then is that it's kind of quite diverse. So there are indeed very experienced ones, very experienced researchers that have indeed pre-registered a lot already. And then others that most likely younger researchers that were not so much in contact with the pre-registration already, that will look into it. But well, I mean, pre-registration is one of the powerful measures to decrease the research bias. So yeah, very nice to hear. And then the last and second last questions, which kind of always belong together for me. So it's how effectively can you find your own research data, and how effectively can you find research data from your colleagues? And okay, wow. So there's quite some people that also have troubles to find their own research data. I've seen that kind of a little bit higher already before, because the way usually it is that people can really have very good control over their own data. But then indeed, when it comes to the data of colleagues, because they have their own system, that it's very difficult to understand what they were doing before. So, but in the end, that's where EQIPD wants to provide help and support to implement systems to basically, increase here, in that case, both numbers. So both questions, you should be able to find your own research data as well as the research data from your colleagues, of course. So, and with that, I'm really at the I'm very much at the end now, and happy to take happy to discuss also anything is how this concept is perceived. Does it make sense to you? Do you think it's completely out of scope? Yeah, any comments welcome.
Yes, Björn, there's a first question asking, do you think pre-registration should be part of the approval process?
Approval process?
No, approval process.
Approval of what? Of articles of
animal experimentation. Just came.
Okay. So basically, should be make it mandatory when you write TVAs. These Tierversuchsanträge. Well, in the end, I believe every measure that we have to increase research data is a good one. So making pre-registration mandatory during the approval process. So right now, to me, it kind of would make sense. But maybe there are some exceptions or freedom of researchers that would be restricted too much or would feel to restrict too much.
Okay, and there's another question coming up in respect to the question you just asked, Björn. So she would love to get some new, or to get the tools how to implement and how to do this. So I know a lot of concepts, but how to finally get them into practice is really the other thing.
I agree with that, and that's exactly why it's also for me so very much interesting to be here at the ZI, because before I kind of theoretically was part of the team theoretically developing the system, but now it's about implementing. And here it's for me it's very helpful to have conversations with other people doing it. For example, Claudia Kurek from the Charité, she implemented the quality system there. It's kind of this exchange which is very important. And so let's meet, let's set up a meeting. But what can I so far? How to implement it? I think it's kind of creating incentives, making it clear why it needs to be done. Always trying to take the researcher on board. That's of kind of at least what I'm trying here at ZI. To say, okay, we need to improve data integrity because, well, we are always searching for it. And just one example where I think it became very obvious. A research project where half a year ago, there was a student working on it for half a year. That student left, saved data in one place. Then a new student came, kind of worked on it. And then, creating this awareness, how difficult it is to find that data. I think that's one of the concepts to implement it. And the other concept is I'm trying with let's talk again in half a year is using the electronic lab notebook and trying to fill it with a lot of information so that it really becomes a resource that, okay, I'm looking for an antibody, I'm going into eLab, I know where to find it, I find the data sheet there, and hopefully that also makes it easier to then document it. And so kind of, create these incentives.
Okay. next question coming up. Have you thought about sharing your system with other quality strategies as reporting platforms about critical incidents in animal experimentations like CRISLAC? I think you are doing that, right?
So exactly. So this quality system kind of shall set the basis, and indeed one of the requirements, which I've not presented, but one of the 18 core requirements is to kind of have a mechanism in place for critical incidents, or in general. And there is the lab series also, and then it kind of depends on the research unit if they want to implement specifically this solution like LabSears. I don't know of this Sears LAS. But in the end, it can be any measure. Like it can also be just the mailbox somewhere where the incidents are dropped in there, or just like a meeting once a week to discuss such items.
Okay, so then there's a comment, not a question. Someone saying that it is very time-consuming to pre-register and to adapt to quality systems. So many experimenters would not like to spend their time for that.
Agree. I agree. It is time consuming, but on the other hand, it's also time consuming to search data. It's also time consuming, or at least I find it very time consuming to read research paper that are biased, because in the end you don't know, and then you read something and you find out again 2-3 years later, or again this Alzheimer example, where 20 years later we find out now that in 2000 or 2006, so roughly 20 years, data were faked. There were over $300 million spent on it. So, well, I mean, with that, we could really hire lots of people that actually take care of that for the researchers. So this is also kind of, that's a cultural, structural change that is also needed in the end. But
of
yes.
Course, yeah. I mean, as you mentioned, Björn, it's kind of, taking them at hand, climbing the mountain, and as you are at the top, it will speed up when you go down again. So, but you have to climb it.
Yeah, absolutely. Yeah. And exactly, especially at the beginning, it's quite time consuming to set the basis, to set the base camp to.
Okay, so I think that were most of the questions. So, there are no more questions, then I would announce that, Georgia will also present the upcoming seminars of our webinar series. And also, you will find the link. So we are recording this webinar, and can be found on the YouTube channel of the 3R Center. And I think there was also a final questionnaire, right?
Yeah, this survey will be presented in the end. So when we close the webinar, there is a window popping up with the survey, and everybody who likes to participate can briefly answer the questions. But of course, just only if you like to.
And it would be very helpful.
Okay, great. So if there are no further questions, then I would like to thank you, Björn. And also thank you for the questions we received. And please take 1 or 2 minutes to finalize the final questionnaire. And with that, I want to close the webinar and wish you all a great evening.